This recording is on lecture 7, which is related to cancer. So the course so far, we have been talking about various ways that humans have been able to extend um, uh, life expectancy through various innovations, right? Either through a medical breakthrough or through domestication of animals. So, we talked about the use of vaccines to prevent viral infection, use of antibiotics to prevent bacterial infection. We talked about organ transplant to fix organ failure, and kind of one of the last frontier that is left to conquer is cancer, right? Thus far, there is no treatment for cancer. I mean, there are surgical options, and some chemotherapy that help people recover from cancers, right? That's possible, but there's kind of no universal cure for all types of cancers. So that's kind of like the last thing that's kind of in the way for humans to have a long life expectancy. So cancer is just one of the many terminal illnesses. There are other terminal illnesses that doesn't have a cure, and what's considered as a terminal illness. Well, typically, there are a wide range of different illnesses that cannot be cured or adequately treated. Okay, so it's reasonably expected to result in death of the patient within a short period of time. So that's basically what's considered as a terminal illness. And depending on the condition and the treatment, the patient might live with terminal illness for just days, weeks, months, and sometimes years. So you can see the time frame is quite a wide gap, right? We don't actually use how long we expect the patient to live to determine whether something is a terminal illness. Okay, so patients are likely to be receiving treatments to help either reduce their symptoms, right, or manage their symptoms. And again, there is no cure for their illnesses. So some examples of terminal illnesses include ALS, right? You might remember Stephen Hawking, right? He was diagnosed with ALS, and when he was diagnosed, the doctor said he's just gonna have a few years to live, but he turns out to be able to live a productive life all the way up to age 76. Other types of terminal illnesses include liver diseases, stroke and coma, and cancer, which is what we're going to be talking about in today's lecture. This is just a clip that talks about Stephen Hawking. If you're interested, you can play it from the PowerPoint slide yourself. So some cancer facts here. About 16% of people die from cancer, and each year about 8.8 million people die from cancer. That is accounting for 1 in 6 global deaths. Cancer affects everybody, but similar to a lot of the trends that we talked about in class, there is disproportionate population that are affected by cancer. About 70% of deaths from cancer occurs in low and middle income countries. So that's like a recurring theme that we've seen throughout the course, right? This was also the case with antibiotic resistance bacteria, as was the case with food supply, for example, and even life expectancy itself was there's a difference between low-income countries and middle-income countries. Top 5 types of cancer killing men are lung cancers, primarily due to smoking, liver, stomach, colon, rectal, and prostate cancers. In women, breast cancer, lung cancer, colorectal cancer, cervical cancer, and stomach cancer. So we know not using tobacco, not smoking, can actually help prevent cancers. Study shows that roughly 30 to 50% of cancers, depending on the type of cancers you're talking about, are actually preventable. A lot of it has to do with our lifestyle and the things that we do. Sometimes it has to do with things that we eat, the things we consume. So tobacco is the single largest preventable cause of cancer in the world. It's estimated that it's responsible for 22% of all cancer-related deaths. Sometimes vaccines can help reduce the chances of cancer, right? We talked about HPV earlier in the vaccine lecture, right? HPV is the human papilloma virus, and that we know is linked to cervical cancer. And of course, there is a vaccination against HPV infection. Hepatitis virus is also linked to liver cancers. So by getting vaccines against these infections, they could prevent up to 1.1 million cases each year. More facts here, right? Access to cancer treatment in low-income countries is, as expected, quite low, right? Less than 30% of low-income countries reported treatment services were generally available, compared to more than 90% of high-income countries. So in North America, much of the population would have immediate access to cancer treatment, but not so much in developing countries. Cancer, because is a debilitating illness that creates significant burden on global economy, right? Not only are people not going to be able to work, right? Sometimes they have to be away from work if they get diagnosed, right, as part of the treatment. So there is a loss of productivity there in terms of the economy. So the economic impact of cancer is quite significant. It's estimated that the total annual cost, yearly cost of cancer is 1.16 trillion US dollars. In terms of palliative care, worldwide, only 14% of people who need palliative care currently receives it. So palliative care aims to reduce the pain of the patient in many cases, right? And want to make sure that they have the medication that they need to reduce the symptoms as much as possible, that they could live their life at a reasonable to have a reasonable quality of life despite of the cancer. Here are some stats that shows you the number of deaths in different age group. So, for example, early on in life, 1 to 24 years, you can see cancer, which is shown here by this light blue color, only accounts for 9% of all deaths. Okay, and then in the next age group, right, 25 to 44, that almost doubles, right? To actually it doubles a little bit more than double, right, than this. So that's 21%. And then as you age to 45 and 64 range, it accounts for 44% of all deaths, right? So this is the most at-risk group of dying from cancer. But the good news is, if you make it past 64, right, you are going to have a lower risk again, right? So it's 65 and over, only 25% deaths are accounted as cause of cancer, right? There are other things that would be more likely to kill you once you make it past 65. So according to these stats, 45 to 64 age group is the group that potentially has the highest risk of dying from cancer. So in Canada, these are some facts specifically related to Canada. This is the lifetime probability of developing cancer in Canada. The stats is a little bit old, right? Actually over 10 years old, but it's still relevant today. In male, you have a 45% chance to develop cancer throughout your life, and it's a little bit lower in female, only 42%. Right? And remember, getting cancer does not necessarily mean you're going to die from cancer. So the lifetime probability of actually dying from cancer in Canada, it's 29% in male and 24% in female. Right? So there is a almost half chance that you're gonna get cancer in your life in Canada, and roughly a quarter chance of dying from it. These are also Canadian stats. It shows you the number of new cases of different types of cancers in male and female. So in male, right, prostate is the most common type of cancers, and breast cancer is the most common type in female. Interestingly, colorectal and lungs make it to the top 3 lists for both genders. And on the right here, it's talking about the estimated cancer deaths by sex. So in male, right, lung cancer accounts for the most death, right, and it is the same in female. Okay, so while you're likely to get prostate cancer, right, as a male, your chances of dying from it, or the number of deaths, right, related to prostate cancer is actually lower. So again, right, just because you get diagnosed by a certain type of cancer does not automatically mean you're going to die from it, right? And that's a good thing, of course. Here we can see that there are different the different provinces and territories, and the cancer rates are actually not the same across Canada. So, for example, you can see Ontario, that's 404.2 per 100,000 population. But in places like Nunavut here, you can see the number is lower, right? So some people say that this might be caused by a difference in population screening. So in Ontario, there are like 76,000 new cases reported annually, but only 80 new cases, right? So that might have to do with the available screening or testing, right, for say breast cancer, right? Some provinces are more well funded than others, and so that might account for some of these discrepancies. So also there is geographic variations, right, in incident rates, could due to modifiable risk factors, right, such as unhealthy diet, right, smoking, obesity, and physical inactivity, right? And also, as I mentioned in the previous slide, the difference in incident rate might also be related to the different programs or procedures for diagnosis and early detection of cancers. So, what kind of approved screening programs are available, right? And what kind of diagnostic service are available. Those tend to change the numbers a little bit. So now we're going to talk a little bit about what is cancer. So some of these things we talked about already in earlier lectures, but let's just go through them again. Right? What is a cell? A cell is the basic unit of life, right? So we as humans are made up of trillions of cells, right? And these cells, they would divide to make more cells, and when they divide, we call that mitosis. Okay, so why do cells divide at all, right? So cell division is necessary for 3 main reasons. Okay, we need the cells to divide so that we can grow, so that we can develop, so that we can reproduce. Right? And some cell divides more frequently than others, right? So it's like skin cells, you replace your skins pretty much in several weeks, but brain cells, they don't really divide at all, right? So depending on the kind of cells you have in the body, some divides more often than others, right? You probably remember I said in the organ transplant unit that if you donate some of your liver, it's actually going to grow back, right, quite quickly. So those cells would have very frequent division. But cells typically cannot divide forever, right? It depends on the type of cells in the body. typically they stop dividing after 40 times, okay? And then they're kind of stuck there. They won't be able to divide anymore. So dividing 40 times is actually a lot of cells, right? So one cell becomes two cells becomes four cells would become 8 cells, right? So if you do 2 to the exponent 40, right, and you can plug that into the calculator, and you can see how many cells would you be able to generate from that single cell in the first place. So lots of cells to go around. After each cell division, okay, the chromosome, the DNA, right, actually gets a little bit shorter, right? And you know, protecting the ends of the chromosomes are something, some special DNA called telomeres. Okay, so think of the chromosome as like your shoelaces, and at the end of your shoelace, there is a little plastic cap that protects the shoelace, right? So that it doesn't fray apart, right? And that's what the telomeres are for. The telomeres are there to cap the end of the chromosomes. So after each division, your telomeres get a little bit shorter, and eventually they will get critically short, and that will send a signal to your cells telling it not to divide anymore. Okay, because if you keep on dividing, you're just gonna cut into your chromosome, cut into your DNA, right? And that could be bad news for your cells. So here is a picture of actual chromosomes, and the little light white dots at the end. Those are the telomeres, right? So over here, you know, we have the original cell, and then it's dividing. So you can see the telomeres shown here by the red segment gets shorter and shorter and shorter. Okay, there is something that we can have in the body to keep the telomeres from shortening. This thing is called telomerase, and that is only present in our sex cells. So like in the sperm and egg, for example, you wanna keep the length of the telomeres the same, so that when you pass it on to the next generation, they are not gonna have critically short telomeres, right? So a cell, its life is basically called a cell cycle. A typical cell spends most of its time in what's called the interphase. So in the interphase, that's when the cell performs normal functions. Okay, so if it's a heart cell, it's going to be beating. If it's a muscle cell, it's going to be contracting, right? If it's a liver cell, it's going to be like detoxifying your blood or something. So they just keep on doing their own function, and they would grow at the same time. So most cells spend, you know, roughly 90% of its time in this interphase, right? And so in the interphase, you're just going to be growing, basically, right? And that's this part of the cell cycle. Okay, so from here all the way to here. Okay, now the cell cannot get too big. If it becomes too big, it's going to be inefficient, right? In distributing nutrients, is not going to be able to perform the function as well. So what needs to happen at this point when it's too big is they need to divide, right? So at the end of the interphase, it is going to undergo mitosis. Okay, it's going to go undergo cell division if it's ready, right? So the cell that divides frequently, like a liver cell, for example, it will progress through the cell cycle faster than cell that divides less frequently. Okay, so sometimes, right, if you take a skin cell, for example, it might go through this cycle in a week or two. Okay, but if it's like a brain cell, then it never really goes through the full cycle because it doesn't really divide. So here is what a typical cell division, typical mitosis event looks like. You have the original cell, which is called a parent cell, and the parent cell will double its genetic material before it splits in two, and then the actual division will take place. So the end results are going to be two genetically identical copies of itself. So basically, it's going to be cloning itself. Prior to the division, prior to the mitosis, the cell needs to check to see if it's healthy and ready to divide. Sometimes the cell is too old, right? Like we said earlier, if this cell's already divided like 40 times already, then it's not going to be fit enough to go ahead with the division, and then it will not. But there are other factors that would prevent a cell from dividing as well, and we're going to be talking about that soon. So here's an actual picture of a cell undergoing mitosis, right? So you're starting with one cell, and eventually it's going to split into two. These dark stuff that you see here, those are actually the chromosome, the DNA. So along the different points in the interface, there are several checkpoints. Okay, these checkpoints are designed to detect any mistakes in the DNA. Okay, so you know, there's the G1 and G2, right? Kind of like your driving license exams, right? So at G1, you're basically going to check to see if the DNA is of good quality. Okay, if it's good quality, then you will go ahead and allow the cell to copy it. Okay, and then once is copied, you want to check to see if there's any kind of mistake or errors in the DNA that you've copied. And if you found any mistake, your cell would try to repair those mistakes. And sometimes the mistakes cannot be fixed, right? It's a permanent mistake, and then that will send signal to the cell, causing it to perform apoptosis, which is basically programmed cell death. So the cell will self-destruct and destroy itself, and it's not going to be allowed to divide. And that's a good thing, right? We don't want cells with errors in them to start dividing and multiplying themselves in the body. So these checkpoints, right? These G1, G2 checkpoints, right? They are actually proteins, right? So the proteins, there are different various types of proteins, and you know, sometimes they are responsible for checking for errors. Sometimes they're responsible for allowing the cells to go faster or slower through the cell cycles. And typically we break them down into two types of regulators, okay, these proteins. Okay, we have growth factors, which would promote cell division. So if everything is good, right, DNA is of good quality, then the growth factors would give the green light, right, for the cell division to take place. And then there's the growth inhibitors that will stop the cell division if the DNA is not of good quality. So they kind of work against each other, but think of them as like the accelerator of a car and the brakes of a car, right? You need to know when to use which, and you know, together they allows you know, productive results to be produced, right? So the proteins, the regulators, these promoters, right? These proteins, they are encoded in the DNA. We learned that earlier, that the instruction to make all the proteins are actually coded within the DNA. So in humans, there are 2 copies of each genes, right? And you might remember we inherit 1 chromosome from each parent, and on the chromosomes there are many genes, right? So you know that's the chromosome from father, that's the chromosome from mother, and on it you would have multiple genes. Okay, so some of these genes might be a regulator, right? Might be a regulator, right? For the cell cycle. So you will have 1 copy from mom, 1 copy from dad. And just to recap for you, right, the DNA that codes for the protein is made up of 4 bases, right? They are represented by these letters A, C, T, G. For the proteins, right, they are made up of 20 building blocks called amino acids, right? And every 3 DNA makes up a codon, and the codon would code for the amino acid. Right, so the chart we used in class previously was a little bit different, but this is similar idea. Right, so if you have AAC, right, that's the instruction for this particular amino acid. Right, if you have GCA, then that would be the code for this amino acid, right, ALA. And so the codes together, it's gonna basically give you instruction to know what amino acid linked to what amino acid acid to create this long proteins. So here is the problem, right? If the DNA mutates, if there is a change, a permanent change in the DNA code, in the codon, in that triplet of DNA bases, then you will likely change the protein. For example, if you have GCA, right? GCA codes for alanine. Okay, GCA codes for alanine. If the C mutates to A, then now you have GAA, and that becomes glutamic acid. So that single change in base might result in a permanent change in your protein sequence, and that could cause the protein to not work properly, right? So if the mutation occurs in one of these regulators, these proteins that are supposed to be controlling your cell cycle, if you mutate them, then the cell might divide uncontrollably, okay? So there are these things called proto-oncogenes. Right? Proto-oncogenes are genes that normally help cells to grow. Okay, so just to clarify that a little bit, the proto-oncogenes are the genes that has the instruction to make the protein which is going to act as a growth promoter. Okay? So when the proto-oncogene mutates, or there are too many copies of it, then it becomes a bad gene. Okay, so this is going to cause the promoter to be permanently turned on or permanently activated. Okay, and that will cause the cell to grow out of control. Right? And when the cell keeps on dividing and dividing and dividing, that's going to potentially lead to cancer. So that mutated gene, the bad gene, is called the oncogene. The proto-onco was a good one, the normal one. Okay, after the mutation, we call that the oncogene. On the other hand, we have something called the tumor suppressor genes. Okay, tumor suppressor genes are normal genes that encode for protein regulators that would slow down cell division. Okay? These are genes that encode proteins that will help repair the DNA mistakes if they found any during the cell cycle, right? Or they're genes that encode for proteins that will tell the cell to die if the DNA mistakes cannot be repaired. So these genes will typically encode for proteins to slow down cell cycle, right? As opposed to proto-oncogenes encode for proteins that would speed up the cell cycle, right? These are ones that will slow it down. When the tumor suppressor gene does not work properly, then the cell can also grow out of control because the checkpoints are no longer functional. You don't have the brakes to stop the cell cycle anymore, and that could lead to cancer. So typically, cancer arises when the mutation occurs in what we call a gain of function in a proto-oncogene. Right? So remember, proto-oncogene codes for proteins that speeds up the cell cycle. Right? That gives the green light to the cell cycle. Right? So think of this as an accelerator in the car. Right? If one of the copy of the proto-oncogene is mutated, that's enough to cause the cancer. Right? That's like having a stuck accelerator, and that's just going to cause the car to speed up very, very quickly. And you know, you don't need two mutations, right? For this to occur, just one mutation causing half of the proteins to be permanently switched on is enough to trigger cancer. On the other hand, we can have what's called like a loss of function tumor suppressor gene, right? So this one is a little bit different, right? Again, using the car as an analogy, you can stop the car potentially with the regular foot brakes, right? Or you can potentially stop it with a handbrake, right? So you need both of them to be completely busted, both of them to have lost the function before your car loses control and spiral towards cancer. So in this case, if you lost one copy to check for DNA damage, it's okay. You still have the other functional one. But if you lost both, then you are going to have uncontrolled cell division. So you could inherit or acquire these mutations, right? Some cancer mutation runs in family, and you can certainly get one bad copy from one of your parents, or you can acquire it throughout your life through exposure to carcinogens. We're going to talk more about that later on. And typically it takes more than one mutation to cause cancer. Sure, if you lost functions in one of the tumor suppressor gene, that could cause your cell division to go a little bit out of control, but there are safeguards in place in the body. There are backups, right, and multiple things to kind of compensate for it. It's when you have multiple mutations, then the cell would not be able to control itself anymore. So if someone inherits an abnormal copy of the gene, then they would have a higher chance of developing cancer. Remember, for tumor suppressor gene, you need 2 hits to get the cancer, right? But if you inherit one mutation, that mutation is going to show up in all of your cells. So it will only take another single mutation to increase your risk of getting cancer. So here it says that is why cancers that are inherited tend to occur earlier in life, right, than cancers of the same type that are not inherited, right? So presumably, it would take 2 hits, right, for someone who did not inherit the error, and the chances of 2 hits in the same cell is actually quite low, and that would take a longer time in life for that to show up. There is also the concept of penetrance, right? Penetrance is the concept that not every mutation is going to be physically manifest. Okay, for some mutations, we have what's called complete penetrance. That means you get the mutation, you get a guarantee to have a problem. On the other hand, we have incomplete penetrance, right? Depending on where the mutation occurs, right? Part of the population seems not to be affected by it, right? So if we say there is a 60% penetrance for this particular mutation, it means 40% of the people will not be experiencing any problem, even though they have that mutation. So as an example, there is something called the BRCA1 gene. BRCA1 stands for breast cancer 1 gene, and it is responsible for repairing DNA damage, right? And if the damage is not reparable, then it will destroy the cell. So this would be a tumor suppressor gene, right? Because it stops the cell from dividing in the event of a DNA damage. Statistically speaking, about 12% of women in the general population will develop breast cancer sometime during their lives. However, if the woman inherited one harmful BRCA1 mutation from their parents, right? So you have 2 BRCA1 genes, right? If one of them has a mutation, then the chances of developing breast cancer by age 70 increases to 55 to 60%, right? So there is going to be a tremendous increase in your risk of getting breast cancer if you have just one mutation in the BRCA1 gene. Okay? So you might have heard of the Angelina effect, which basically the actress, right, Angelina Jolie, she undergo double mastectomy because her genetic testing shows that she has a BRCA1 gene mutation, and so she didn't want to risk getting breast cancer later on in life, so she opted to perform a double mastectomy. So accelerated uncontrolled cell division will result in the formation of a tumor. We call the tumor a neoplasm. And tumor is not necessarily cancerous, right? Some tumors are benign. Benign means not dangerous, not harmful. And typically you can just remove them with surgery and there will not be any further complication. If it's cancerous, we call that a malignant a neoplasm, and that will get progressively worse and are dangerous to health. So typically you have a normal cell here, and then you will have some kind of mutation in the DNA. either a gain of function of a proto-oncogene or loss of functions in the tumor suppressor gene, and that will cause the cell to divide uncontrollably, and it will progress through different stages of cancer, and it might even metastasize, spread to other parts of the body, and ultimately result in death. Right. So most of the treatment aims at preventing the illness from going to the to this stage here, right? If you try to destroy the cancer cells early on, then you have a higher chance of surviving. So here are some of the hallmarks of cancer, right? They have cell sufficiency in growth signals, which means they no longer require external signals to divide. Okay, they don't need the growth promoters to tell them to divide. They will just keep on dividing no matter what. And to compound that effect, they are insensitive to anti-growth signal. Okay, even if you tell them to stop, they don't care. Right? They will just keep on going. Right? So these two things together, it basically means that it's green light for them all the time. Okay, they see a stop sign, they see a red light, they don't care. They just kind of keep going. All right, so these two are hallmarks of all cancers. They have the ability to evade death. Right, essentially they become immortal. In fact, it's commonly known that if you take cancer cells from a patient and out of the body, and then you put them in a petri dish, right? They could grow on forever, right? They are immortalized. They have the ability to form new blood vessels, and that's kind of important because when the cell kind of keeps on dividing and become this big mass of cells, they need a lot of nutrients. Okay, so with the existing blood vessels in the tissue, that typically will not supply the entire tumor. So by having the ability to create new blood vessels that goes into the tumor, or that dedicates a supply to the tumor, they are able to ensure they have an adequate supply of nutrients. They have limitless replicative potential. They no longer become senescent. Senescent is what happens when the telomeres becomes critically short, and that usually would signal the cell to stop dividing. But they don't care. Okay, they have a way to fix the telomeres to keep them long. Right, just like our sex cells, they keep the telomeres long, and they could keep on dividing and dividing forever. And lastly, they have the ability to invade and spread, metastasize to other parts of the body. So these are the 6 hallmarks of cancer. So a recent study reports that 66% of all genetic mutations that cause cancers are simply random errors. So that means it just happens, right? There's no avoiding it, right? In the process of copying DNA, and there are like billions and billions of those ACTGs to copy, it's inevitable that there is going to be some kind of error when you transcribe them. Okay, so it's just going to happen, right? While random cancers are part of the picture, it's important to remember that lifestyle and hereditary factors are also still an important part of what's causing cancer. So this notion of nature versus nurture talks about whether you are born with the risk of getting certain cancers, or is it something you do in your life, right? That increases your risk of doing it. So nature versus nurture. So the American Cancer Association states that more than 50% of all cancer deaths can be prevented, and we kind of talked a little bit about this earlier, right? So substances and exposures that can lead to cancers, they are called carcinogens in general. Carcinogens do not cause cancer in every case all the time. You need to be exposed to it for a certain period of time in some cases, or you need to be exposed to it to a certain level before you are going to have significant risk. And sometimes it is depending on how potent the carcinogen is. Sometimes just a little bit of exposures would increase your risk tremendously. Other times, not so much. So substances labeled as carcinogens might have different level of cancer-causing potential. Like I said, right, some might cause cancer only after prolonged high exposure, right? and sometimes it depends on other factors such as how they are exposed to the carcinogen, right? Did they ingest it? Did they just touch it, or were they just in the immediate area of the carcinogen, right? So it depends on what you're talking about. Those things would matter. And the length and intensity of the exposure, and as well as the person's genetic makeup. Okay, some people are more prone to be affected by these carcinogens than others. So, how do you identify carcinogens? Testing to see if something can cause cancer is kind of a difficult thing, right? Like, you cannot just expose regular people to it and see if they get cancer, right? That would be extremely non-ethical, right? And you know, no one would want to participate in those studies, right? So much of the studies of carcinogen actually are drawn from lab studies, right? Perhaps using animal models, as well as epidemiological studies. So just by looking at the population and see, you know, what kind of cancer a population gets, and are those individuals in certain populations more prone to getting certain types of cancers? And if they are, then, you know, are there something certain lifestyles that are unique to that population that might be contributing to that increased risk? So these are the kind of things that people look at in epidemiological studies. So one example of that was the Hinkley groundwater contamination. So that's where the town is, Hinkley, in the States, California. And what happens was, you know, this organization, this company, the PG&E, they use something called chromium-6, which is a cheap and an efficient rust suppressor to use in their compressor station for natural gas pipelines. Okay, so to transmit natural gas in a long distance, every now and then they have to go into some of these a compressor to increase the pressure, so that they're going to be able to keep on going forward. And so to prevent rusting in these suppressors, this company uses chromium-6 as a basically anti-rust agent. And what happens was that from 1952 to 1966, this company dumped about 370 million gallons of these chromium-tainted wastewater into the unlined wastewater spreading points around the town of Hinkley, and it turns out that this chromium stuff, chromium-6, is cancer causing, is a carcinogen, right? So in 1993, there was a legal clerk. Her name was Erin Bukovich, and she was looking into this situation and investigating the health impacts of the contamination. And that resulted in a class action lawsuit about the contamination, which was settled in 1996 for 333 million. So in this story, right, basically it's a epidemiological study, an example, right? They found out that this town, people in Hinkley, were getting abnormally high rates of cancers, and, you know, nobody seems to know why. And then when they dig a little bit deeper into it, they found out that because they were exposed to the chromium-6 in the drinking water, essentially. And so, you know, that linked, now we know the chromium-6 is a carcinogen that is pretty potent at causing cancer. The International Agency for Research on Cancer, or the IARC, is part of the World Health Organization, and one of its major goal is to identify basically what causes cancer. So according to them, the carcinogens are classified into different groups, right? Group 1 being non-carcinogenic to humans. So these are the things that are going to increase your risk of getting cancer substantially, which includes things like smoking, exposure to solar radiation. All right, so that's why people say you have to put on sunscreen when you go out, so the UV is not going to damage your DNA. Particularly, we have a hole in the ozone, right, and that is what's causing the radiation, making the radiation to be carcinogenic. Drinking alcoholic beverages and eating processed meat, so like sausages, bacon, ham, that kind of stuff, right? So these are things that are known to be carcinogenic to human, right? And then as you go down the group, the severity decrease, right? So group 2A probably gonna cause cancer, but you know, there are not enough evidence, right? So example would be things like, you know, frying, exposure to steroids, right, red meat, right, and then in 2B, things like coffee, gasoline. So it's actually kind of interesting, right? In some states, in the U.S., they actually, like on your Starbucks coffee or something, Starbucks coffee, they would have to say potentially are carcinogenic, okay, because of this recommendation. And you know, group 3 are not classifiable. Like basically, we don't know much about it in humans, and we haven't really tested them on animals as well. And for group 4, probably not carcinogenic. So you know, if you think about having a barbecue at the on a weekend, right, on a hot summer day, you know, drinking some beers, eating some sausages, right? So in this picture, there are like 5-6 things that are potentially carcinogenic, right? So I guess like a lot of modern life living places to substantial risk of so can we prevent cancer? Well, there are some pretty basic suggestions that are out there. Increasing physical activity, maintaining healthy weight gain, right, avoiding tobacco, and so on and so forth. So these things, while will not, you know, will not be 100% make sure that you don't get cancer, but you know, you could reduce the risk substantially. So let's talk a little bit about you know, how evolution might have played a role in cancer. You know, if you remember, we talked about evolution before. The main drivers of evolutions are basically mutations, right? And sometimes they're good mutations, sometimes they're bad mutations, and the principle of natural selection. So if you have a mutation that's favorable and that creates some kind of survival or reproductive advantage, those mutations will tend to prevail in the population. On the other hand, if you have mutations that are unfavorable, if it causes some kind of disadvantage in survival or reproduction, then those will tend to go away in the population. So the puzzling thing here is this. Cancer are caused by mutations, and these are pretty negative mutations because they, of course, decrease your chances of survival. So how is it that evolution did not take them out? How is it that natural selection did not cause these harmful mutations to disappear in the population? So the theory says that what if there is a mutation that affects survival in post-reproductive stages of life, right? So what that means is, what if there is some kind of mutation that has benefits early on in life, but then has the opposite effect later on in life, right? So something like that could be preserved. So evolutionary theory holds that aging is a consequence of declining force of natural selection with age. So factors such as accident, predation, well, this doesn't really happen anymore, but like, you know, in the past, it's quite possible that you get eaten by a lion or something, for example. Infection, starvation, right? So these factors limit lifespans of most species, therefore depleting natural population of older individuals. What this is saying is there are other factors that are much more likely to cause someone to die later on in life than the mutations themselves, and so consequently, there are generally fewer old survivors on which natural selection can act on to eliminate the genes, right? Because they have late acting deleterious effect. In other words, if the mutation is going to cause negative impact later on in life, they might not get weeded out by evolution as easily because the individual is going to die before any of those effects shows up. So, of course, in modern life, right, because we are living longer and longer, right, due to the increase in longevity, right, life expectancy, that's why now in modern days we are seeing more and more cancer cases because of these late-acting deleterious effect. Natural selection cannot eliminate genes that promote early life survival, but strangely promote late life debility. Right, so this kind of paradox where you have early on benefits, but then disadvantage later on in life. This is called antagonistic pleiotropy. Right, so one example of that is in male. High testosterone level in males can actually increase fitness early on in life, right? So, you might have bigger muscle mass, you will be stronger typically, and allow you to have higher survival and fitness. But as a result of that, it turns out if you have a high testosterone later on in life, you are more prone to getting prostate cancer. So this is a classic example of how you can have early on advantage in life, which natural selection would be keeping it, and by the time it shows up later on in life to be having a negative impact, you would have had a child at that point already, or kids already. So you would have already passed on that gene, right? And as a result, you cannot select against that. Another example is cellular senescence, right? The aging of your cell and not be able to divide anymore. So that acted as some kind of benefit early on in life because it actually suppresses cancer. But actually, later on in life, it was discovered that the cellular senescence causes aging, and of course, aging is related to cancer. So those are two examples of antagonistic pleiotropy. So are there any treatments? Well, there is kind of like a three-pronged approach to treatment for treating cancers, right? Surgery, if you can remove it. Radiotherapy, if you can use radiation to destroy the cancer cells, or systemic therapy, which is more of using chemotherapy, for example, right? Medications, right? Immunotherapy, using your own immune system to target the tumor cells, or you can use hormone therapy. So these are some examples of each, and you can look through them yourself. And the goal of the treatment is, of course, right, to eliminate cancer if possible. And sometimes that's not a viable option, right? Then you might be looking at shrinking the tumor, therefore extending the life expectancy of the person, right? You want to prevent it from spreading it to other organs, and you do want to minimize the symptoms, such as pain, as much as possible. So you can give the treatment as an IV, right? Sometimes just pills. You can have an infusion pump that delivers the drug periodically, or done through injections. And that's it for the lecture. Thank you for watching.